I'm super happy to be here and seeing so many friendly and friends' faces. So yes, today I'm going to talk about some work that we did at SpiNNaker with me with colleagues at SpiNNaker and with collaborators from specifically Egidio and Claudia. So this was very well organized for this talk to be right after theirs because we use a Cerebellum model built by them to try and run it on neuromorphic hardware. And neuromorphic hardware is brain-inspired hardware that tries to make some assumptions about the types of neural networks that you want to run in order to accelerate their execution. So neuromorphic hardware wouldn't run very detailed neuron models. It would focus on point neuron models in the hopes that you can run large numbers of them. In the case of SpiNNaker in real time, in the case of other platforms in accelerated compared to real time. So why focus on the cerebellum to try and simulate this brain region? Well, as Egidio said, it's a very well studied region, hopefully. And it's very much involved in motor learning and coordination. So we'd like to understand more of that and actually potentially put it to use. It's also a region of extremes. So it makes it quite a fun challenge to try and approach on neuromorphic hardware. So it has some of the most numerous cell types in the brain, cerebellar in the form of cerebellar granule cells, and some of the most innervated cell types in the shape of Purkinje cells. With some Purkinje cells in humans estimated to have around 100,000 incoming synapses per cell. From an engineering perspective, the cerebellum is quite nice. Despite the list of extremes that I mentioned, it's somewhat built out of stereotypical modules. So if you could do module, then you could scale it up by simply having more of them if you have more compute resource. And it's also very interesting, besides the motor learning and coordination, for the fact that it receives and produces a lot of signals that integrate widely throughout the brain, including receiving connections from cortex, brainstem, and periphery to be able to produce useful outputs. So the team in Manchester produced a computer called SpiNNaker that I'm going to talk about soon, with really two goals in mind. One was to explore whether we could accelerate our understanding of brain function at a more fundamental level by essentially using very massively parallel computing resources. And the other question is, could we take information the other way? Could we get information from our ever-growing knowledge of how the brain computes and how the brain predicts to create more efficient, parallel and fault-tolerant computing platforms? And this second one has a more obvious engineering application in robotics, where a cerebellar model like Egidio, Claudia, and Alice showed, where that you could put that to good use in maybe in a smaller scale, but in a real robot, if you had sufficient compute resources on the robot itself. So the talk is basically divided into 4 parts. It's a very quick presentation of the challenges of the Cerebellum model, a quick overview of the SpiNNaker platform for those of you who've not seen a talk on SpiNNaker before, which is fine. One of the main topics of this project, which was activity validation, followed by the communication profiling, because you may have noticed in the title of the presentation, it's towards a real-time neuromorphic cerebellum. So neuromorphics promise acceleration of spiking neural networks. This presentation also covers why this is not immediately guaranteed. It's not a guarantee that you'll get acceleration for your neural networks if you go for neuromorphics. In any case, the Cerebellum model. So the Cerebellum model that we use for this work is a biophysically constrained model generated with the tool that Claudia presented earlier. It's almost 100,000 neurons and 4.2 million synapses. The challenge for this model was to firstly validate that this model could be run on SpiNNaker. I may have time later to discuss why this is needed. Mainly because SpiNNaker, in order to be power efficient and fast, but also be built cheaply, does not have floating point unit. So it relies on fixed point arithmetic for all of the operations that it does. And the second part that hasn't been realized yet, this is hopefully something for the future, is to optimize the model to allow for hard real-time simulation. The communication profiling is very important for this step to actually happen in the future. So the challenges for the model are the Purkinje cells don't have quite the same fan-ins or are not as innervated as real human cerebellum cells, but they still have quite high synaptic fan-ins. Activity can arrive in synchronized fashion, which poses problems for the communication fabric on the machine. And for the numerical simulation of the leaky integrated fiber neurons, there's quite a high dynamic range of weight values, and the cell parameters are different for all the cell types because, of course, they are. They all need to serve a different purpose in the model, and the model needs to be run at least in 0.1 millisecond simulation time steps due to accuracy of simulation. If you go any larger than this, then your simulation is not accurate. This is the general overview of this neuromorphic cerebellum. What kind of compute resources do we have? Well, the essence, the building block of SpiNNaker is its chip, and its chip has 18 ARM cores with some instruction and data core local memory. So each processing unit has some local memory that it doesn't share with anyone else. A very large, if you can see my cursor, a very large SD-RAM wire bonded on top of the chip. So it's a 3D package. That one's 128 megabytes. It's quite large, but very slow. That's where we store synaptic information. And very importantly, a router. Neurons are nothing if they can't communicate their spikes to nearby chips. So this is the basic building block of the SpiNNaker machine. This router is what you could call the magic sauce of SpiNNaker, as it can consume incoming spikes and produce multiple outward spikes in a sort of analogy to how an axon works on a neuron. So you can have single spikes that get propagated onwards from source to target, but also you could have single spikes that are multiplexed to arrive to all of the different targets that it's supposed to arrive at. And this is called multicast routing. And this is quite special for SpiNNaker. And this is what essentially makes all of the magic work, as it were. So in Manchester, we built this SpiNNaker machine, which is in total 1 million, it's actually over 1 million ARM cores. And we aimed for this. So the general use case for this platform is to be subdivided, for tasks to be subdivided into individual boards, as you can see here, or you could use the entire machine. Florent will talk after this presentation about how you can access this system online from your computer through a browser. Of course, having dedicated access to a machine can allow you to do more things, but that's somewhat beside the point here. So this is SpiNNaker, and we used, for this Cerebellum model, we could fit it on 3 boards, but we'll talk later how this could be done in future to allow for faster simulation. So activity validation, because SpiNNaker has some components, has made some assumptions about how neural networks, spiking neural networks would behave. It has a certain design spec, and the Cerebellum model somewhat stretches this design spec. So we needed to make sure that whatever model we run on SpiNNaker produces the behavior that we expect. So the current cerebellar model, originally written in Nest, has been translated in PyNest, sorry, has been translated to Pine, and the same script that runs the simulation on SpiNNaker is also used to run the simulation on Nest. Some minor differences, some minor setup differences have to be accounted for when targeting either system, but this is taken care of even in the current implementation that, again, Florent will talk about in a bit. Some of the features that we looked at quite a wide range of things between these two simulators to check that everything was behaving correctly, from checking that the weight is actually represented correctly, and the activity, the peak activity in the network can actually be represented on SpiNNaker correctly, as well as higher level analysis supported by Elephant, the electrophysiological analysis toolkit. And I can sort of show you vaguely what at the top level this would look like. You can have a look at the activity, the raster activities, and the peristimulus time histogram pairs for each of the populations in the model, left with SpiNNaker, right with Nest. And you can immediately, just by looking at the bins, you can tell that there's a difference between SpiNNaker and Nest. They're not identical. And this is of course expected. There are many differences between SpiNNaker and Nest that would lead to differences, but overall, the differences can be accounted for, and they're not large enough that we can't say that the model matches Nest in this scenario. And I hope, Florent, will you be showing some? Hopefully, you'll be showing some of what the analysis looks like and or how it can be run. After activity validation, which was at the single cell level and then at the large scale level of the entire network, at the large scale network level, we also needed to look at how the activity propagates through the network, because this is important on SpiNNaker. What we looked at was essentially how many packets, so spikes, would arrive at each core in the worst case time step for a wide range of activity patterns put at the input of this model. And now on the right, you can see that in some of the worst cases examined here, some of the cores, mainly those in the molecular layer, so stellate and basket, but also Purkinje cells and Golgi in the granule cells, and the granular layer can receive upwards of 1,400, 1,500 spikes, packets in the worst case time step. But a previous study on that managed to run a real-time cortical model on SpiNNaker found that SpiNNaker was actually capable, with the architecture used there, to consume approximately 20 packets per core per time step. This is once again in real time and with a time step of 0.1 milliseconds. So so something has to change for the Cerebellum model to be simulated in real time on SpiNNaker. Mainly, some of the cores need to be able to some of the populations need to have cores that can process about 20 to 50 times more spikes in the worst case time step. What we suggest is a split synapse in neurons simulation paradigm, where in the current way of doing things on SpiNNaker, each processing core simulates both the synapses and the neural dynamics of the neurons. Taking it a step further, you'd have synapses that rest on one core, on cores distinct from the neuron soma, essentially. but scaled up. The previous model could only have synapse cores that would be on the same core as a neuron. We can't have that for a cerebellum. So future work will have to focus on being able to place neurons on some chips with tens, potentially hundreds of synapses on other chips. This kind of model could also accommodate for some of the models that were discussed in the previous session regarding multi-compartment models for SpiNNaker. So currently SpiNNaker does not officially support multi-compartment models, but such an organization may allow more realistic simulations of neurons, providing that a thorough numerical analysis is performed on these models to make sure that they're validated and working correctly. So in summary, what I've been talking about is the cerebellum, so a brain region heavily involved in motor control and learning, and trying to map this biophysically constrained cerebellum model onto this neuromorphic hardware. And we found that in the current case, a fixed point arithmetic and 16-bit weights are sufficient to model this network. Not 100% yielding the same results as NEST, but close enough. The system can do more. SpiNNaker can do more. So a bespoke software system in the future should be able to cope with 20 to 50 times larger peaks in activity per time step, which would allow such a cerebellum model to be run in real time. And thus, if you could simulate such a cerebellum model in real time, you could analyze its behavior over very long timeframes. You could simulate days in days rather than simulate days in weeks to months. And this could open up further understanding about how the cerebellum computes things, but also if you can run it in real time, then you could put it in robots, and that would be really cool. Finally, robots that can balance themselves and walk normally, for example. All of these results, the results of this work, are freely accessible through EBRAINS. We've tried to make sure that all aspects of this work are transparent and reproducible and accessible. So if you were to, for example, scan the QR code, you would be taken to the repository, the EBRAINS repository that contains all of the results and analysis and software used for the study, as well as Docker containers that would allow you to run NEST or SpiNNaker. If you had a SpiNNaker board to use, you could immediately use Docker containers, so essentially virtual machines, to run your experiments there and see whether you get the same results or not. And with that, I conclude, and I thank you very much for your attention and for your invitation. Florent, would you like to take over?
Yes. Andrew, how much time?
You have 20 minutes left, including questions. So if you can finish a bit quicker than that, then that will leave some time for questions at the end.
Okay. Let's try to share my screen. Okay, do you see something?
Yes.
Okay, great. Okay, so basically I've taken the model and the simulation that Petrut and the University of Manchester and have developed, named Spencer, and I tried to I will try to show you how one can upload this project or your code or any model in order to on EBRAINS tools in order to compute your model using SpiNNaker or BrainScale's computing machines. So basically, when you ask or when you request an account for using neuromorphic computing system, you will have access to 2 machines, BrainScales and SpiNNaker, that Petrut already have explained to ourselves. And this demo only shows how to use the SpiNNaker machine in order to run the Cerebellum model. And the first step to do it, you will have to request an access, and this is basically very simple, as it is done through 3 steps. And the first is to contact the EBRAINS support, and all the other steps will be explained to you by the support team. Basically, there are 3 ways to run a job, to submit a job using EBRAINS neuromorphic computing tools. The 2 first ones are using the Job Manager, which is a tool that can be used through a web interface or a Python client. And the third one is simply using the SpiNNaker interactive mode. Perhaps I can show you how the Job Manager can be used, and with a live demo, please. Okay. So when you request for an access to a SpiNNaker machine, you will have a templated collaboratory that will be developed for you, and you will have access to this kind of web interface. So on the left menu, you click on the Job Manager, and you will be logged in, and also you can add, you can click on these two plus buttons, then to upload your code. So first you select the machine you want to use. Here is SpiNNaker. You can also want to describe your model through a Python editor, or you can also just describe your model using your Git repository or a zip archive. I will use the zip archive for this demo. The third field concerns the Python script that is supposed to be used in order to run your model. Here, the Python script and the required parameters. This is the location of the Python script within the Git repository, and all the things all the following things are the required parameters. The fourth field concerns the hardware configuration. This is a little bit tricky, as the documentation is not very clear about that. So you may need to investigate this field if your model does not work very well for this in the first run. And the last field concerns the tags you can add to just try to retrieve your job within the job scheduler. Okay, I think everything is good for me. Let's just submit our job here, which has the status of submitted, and will be running soon. So Okay, I can show you the any results that have been already done. When your job has finished, you can just click on a certain button, and you will have a list of all generated files that have been generated by your model, and also the complete log. Then you can open and just read it if you have some time to do it. This is the code you already you used, the command line, and the log is also written here. So this is the way to run your job using the Job Manager web interface. Just up. And this was the first. Okay. The first way to do it. You can also do it using a Python client. Here's the templated notebook, Jupyter notebook, that you will receive when you requested the SpiNNaker access through the EBRAINS support. You have some things to install, the tools that will the client, the entire client, and the client is created here. So you can just ignore all these kind of code as it is brought to you by the support in order to give you a very quick access to SpiNNaker Machine. The important part is here, where this is the function that will submit your job to the job scheduler. The source concerns the GitHub repository we already have uploaded. The SpiNNaker Machine, the collab idea that is calculated here, and the config parameters concern the hardware configuration that we already have, that we already saw, and the command line is the same. So we can just submit it another and just check if the job has been submitted. Okay, you can see that the previous job is running, and the one we already submit is queued. We can go further. This is the we can already check the job status, and I already have done using another job submitted yesterday, and you can just check the dictionary that the main informations including the total log resulting the job, quite long, and you can also download your results, and it will download your results. Okay, using this function, it will download your results within a subfolder named with the job and the job ID, and just and you can explore or execute some something in order to extract data and from this raw data you already download. Okay, the third operation, the third way to use the SpiNNaker machine consists in using a direct access to the SpiNNaker through a terminal. On the left menu, you can click on the SpiNNaker submenu, which gives you access to this to a new Jupyter lab that will be generated. I think, yes, okay, it's working. And to give you access to your personal space within this SpiNNaker machine. Okay. Petrut and me have already have downloaded the code we will use for this demo, and we already have also a container environment in order to get all packages already installed. So let's activate this environment, and we can just where is the script? Okay, we can just copy and paste the scripts, and don't forget the Python call. And as you can see, all the model will run, and you will have the generated results within this Jupyter lab. Okay, this is mainly the three ways in order to upload and submit your model, your code to the SpiNNaker machine, or but I didn't have tested yet to the brain scales machine. There are some requirements in order to format your format your code properly. One of them is to have a single Python script. The default one is named run.py, but you cannot call multiple Python scripts in order to get your simulation run. And you also have to fill to give to the job scheduler a zip file, or give to a repository, or either a single Python script file in order to make things work. There are some little bit tricky things to do when you notice that your model or your script doesn't work during the first submission. Perhaps you have to add a SpiNNaker configuration file, and just or have to edit it to have access to give access to SpiNNaker to your model properly. There are small things that are not running out, small bugs to do to fix for the next versions of the Job Manager tool. And one of the most important is the lack of technical documentation. For example, the hardware configuration, the number of the versions of SpiNNaker tools, which can be very time consuming in terms of investigation. But the support is here for you to help you make your model work. And I think it's done for me. As we already have seen, there are three ways to submit to adapt your code to the SpiNNaker machine. The Job Manager and the Python client are batch mode. It means that you need to submit your job to a job scheduler, and the interactive mode, when you have direct access to the SpiNNaker machine. There are a lot of pros and cons, but I think they are pretty easy to use concerning the Job Manager interface and the Python client. So thank you. And